Good day, everyone. Welcome back to class. I trust that every one of us we are doing great. Thank you for your support. All my returning subscribers will say thank you for your support. And if you are coming across this channel for the very first time, this is where we learn biochemistry in simple and plain English. So you are welcome on board and enjoy the ride with us. Please do subscribe and share this video for other people to also enjoy what we enjoy here on this channel. So what we want to discuss today is protein sequencing. In our previous class, we've discussed about amino acid, we've discussed about protein, and for both classes, the most important thing that will be of help to us today is that amino acids serve as building blocks of protein. When we have a particular protein, the composition of that particular protein are amino acid. Let's take this example. This are a bracelet. This bracelet is made up of beads. So it is the arrangement of those of the beads here that makes up these bracelets. Let's take our amino acids to be the beads that are here. So this one is one amino acid, two, three, four, five, six. So when these amino acids join together, they now give us protein. So when these beads are joined together now, they give us bracelets. So when amino acids are joined together, they give us protein. So if you now say you want to sequence protein, mean that you want to arrange it. What we mean is that the amino acid that is in this particular protein is what we want to deal with. We want to identify them, we want to arrange them. We want to know everything about the so-called amino acids that made up this particular protein. In sequencing protein, it could be of two types. You might have a particular protein of interest that you just want to sequence, or you might be given a particular solution that contain particular protein of interest, and they will tell you to isolate your protein from that solution and now sequence the protein. Or they might not even give you a solution containing protein. They might give you an organ that contains protein. They will tell you that you should bring out the protein of interest from that particular organ, then from which you will now sequence your protein. So now let's take them one after the other. Let's start with the scenario that you have a particular organ. Let's say you have a liver organ and you are asked to isolate protein from the organ. So how are you going to go about that process? First, the liver undergo homogenization. And what is the essence of this liver undergoing homogenization? It's just for us to break open the cell. Let's say, for example, you want to cook, let's say, jollof rice now, and you have your tomatoes, you have your bell pepper, you have your red pepper. The first thing you want to do is to blend those pepper to give you a smooth pepper mix before you now continue to cook your jollof rice. So here we are not blending pepper. What we want to blend is our liver to give us a paste. So we are grinding the liver to give us a paste which we are going to use for other processes. So remember, when you want to grind your pepper mix, you will add water to your tomatoes, your bread, the red pepper and other peppers. You add water to make the blending stress free and smooth. So in our case, we are not adding water. What we are adding is what we call buffer. And as you know, buffer solution help to resist change in pH. So your organ of interest, which is the liver, will now plus buffer. So we have your liver here and buffer solution. Let's take, for example, we are using a phosphate buffer here. So most of the time, 1 gram of your organ of interest and like 5 to 10 mil of your buffer is what you will use for homogenization. But this actually depends on the quantity of what you are looking for, or what you are looking for, what you want to work with. Then you homogenize this. The paste that you got here is what we call our homogeneity. Now, what are the things that you can use to achieve that paste solution? One, you can use an homogenizer. You can use mortar and pestle. You know the ones we use for our practicals. Another thing that you can make use of is that you can make use of blender to blend your organ of interest to give you the paste that you want. Another thing you can use sand to break open the cell, and a lot of other methods that you can make use of. All those methods, the point that we want to bring out is that we want our organ of interest to be in a paste form. And when we achieve that, that means we have been able to break open the cell. Next thing we are going to do to our homogenate is that we would now centrifuge the homogenate. It's like you have a mixture of something you want to separate. So that is what centrifugation will do for us. So from homogenization, we move on to centrifugation. And in this process, we just want to separate our paste, which is our homogenate, that we're able to get from homogenization method, where we make use of either homogenizer, either a sound, or either blender, butter and pestle, etc. like that. So our homogenate will now come here is what we are using for our centrifugation. So that paste that is like this, let's say this is your paste. After you've blended everything here, the liver and the buffer, you've blended everything, you got your homogenate, which is our paste that I'm referring to. After this one is completed, what we are going to have now is separation will have occurred. So here we will now have some. There will be some here, and there will be some here. Have the supernactant, which is the one at the top. So we have the supernactant, then we have the pellets. So we are getting two things after our centrifugation. Some will be at the top, some we have some residues at the bottom of your tube. So the ones at the bottom of your tube is what we call the pellets, and the ones at the top of your tube is what we call the supernactants. So we'll take another tube, you will decant, you will pour the supernactant into it. After this centrifugation, the next step is sorting out. But this step is not always common. This sorting out step is not always common, but I just want you to know that we also have sorting out. And what happens in this sorting out is that it helps in reducing the solubility of the protein that is in our solution. It helps to reduce the solubility. It's like in a solution, you have proteins that are already mixed with the liquid of the solution. In this case, is our buffer solution that we use in the homogenization process. So our protein of interest is already mixed with that buffer. So we now added the salt solution to it. So both the salt solution and the protein solution are now fighting for who will get the attention of the buffer that is there. So what the salt solution will now do is to try to restrict the movement of the protein that are in our solution. So these will make them not to be able to flow well in that solution, and they will gradually be coming together, so leading to precipitation. So that is just how sorting out work. And the salt that will make use of for in sorting out is ammonium sulfate. So after that is done, the next step that we are moving on to now is chromatography. The principle behind chromatography is that it works with two phases. We have the stationary phase and we have the mobile phase. For the stationary phase, they, as the name implies, stationary, they don't move. And for the mobile phase, they can move. Now, for the stationary phase, it can either be a solid phase or a liquid phase. But for your mobile phase, it can either be a liquid phase or a gaseous phase. So now, the rate at which your sample will be separated, the rate of separation is determined by the interaction of your sample with these phases. The way your sample interact between both the stationary phase and the mobile phase of the chromatography will determine the rate of separation of that particular sample that you are working with. That's just how chromatography works. So we have different type of chromatography. We have the column chromatography, we have the paper chromatography, we have the thin layer chromatography, we have HPLC and the rest. So it helps us to separate, and also they're helping us to purify our proteins the more. So now, after our chromatography process, the next phase we are now moving to is electrophoresis. This is the process of separation using electric charge. Using electric charge. So that is what we do here. So electrophoresis is just the reaction between your sample of interest that was gotten from chromatography and electric field. The interaction of the sample in that electric field will now determine how the sample will be separated. So after the electrophoretic process now, the next thing to do is now to sequence our protein. The first step in protein sequencing is breaking down of protein into small proteins. So the small proteins are what we call peptides or fragments. So we have a particular protein that they give us that, okay, take this protein, sequence this particular protein. Remember our structures, our previous class on protein, where we talk about different structures of protein. We said we have four structures. We have the primary, the secondary, the tertiary, and the quaternary. That's saying that proteins are very large molecules. So for us to work with them and be able to know what's made up of these protein, we have to first break them into small pieces to make our work simple. So now, breaking these proteins into small pieces, which we call fragment or peptide, is what we call protein fragmentation. So that is the first step in sequencing of protein. So how do we achieve this process? It could be of two types. One, we can use enzymatic method. Two, we can use chemical method. Now, if we decide to make use of enzyme to cut the protein, what are the enzymes that will help us to serve as this function, this enzymatic cleavage function? One, we have the trypsin. Two, we have the chymotrypsin. So this enzyme can help us to cut, to cleave proteins into peptides or fragments. Now, how will these enzymes perform their function? They cannot just go ahead and be cutting at any site they desire. No, they have specific amino acids that they work with. They will only go and cut at that specific amino acid. For the trypsin, it can only cut a protein into peptide wherever it is the amino acid lysine and arginine. So no matter how big your protein structure is, if you introduce your trypsin enzyme and it did not see lysine or it did not see arginine, it will not help you to break that protein into peptides or fragments. Do you understand that explanation? So they only cleave, they only break your protein at whenever they see amino acid lysine, arginine, that ahead. They say, oh, this one na my guy, na my party. Let me come in here. So that is where they cleave the protein structure to now give us small peptides. The second enzyme, which is the chymotrypsin, if this chymotrypsin will help us to cleave our protein to give us peptides and fragments, it will only cut at the amino acid one, phenylalanine, two, tyrosine, three, tryptophan. So anything outside those three amino acids, chymotrypsin will not cleave your protein. So if you have a very large protein and you introduce your chymotrypsin and there is no tryptophan, there is no tyrosine, there is no phenylalanine, this enzyme will not cleave your protein. Let's move on to the second method, which is the chemical cleavage. So what happened is that instead of using enzyme that we use in the first method, now we are making use of chemical. And the main thing that we are after is just to help us to cleave this protein to give us peptides or fragments. So the chemical that we are using under this method, which is the chemical cleavage, is cyanogen bromide. Also, like the first step, which is the enzymatic cleavage, this chemical also have its own crush that it can only work with. So if you introduce this cyanogen bromide into your protein solution and there is no methionine there, this cyanogen bromide will not cleave anything. So it only cleave a protein at the methionine amino acid point. So if there is no methionine, this chemical will not cleave the protein. What is now also common to them is that they cleave at the C terminal of your amino acid or your protein of interest. Now they cleave at the C terminal. Let's do a quick let's branch a little and explain what are C terminal and our N terminal. From our previous class about amino acids, their structure and the rest, in that video we explained in a simple term how you can draw your amino acid. We said your amino acid always have number one the alpha carbon, which is at the center. Then to your right hand now we have the COH, which is the carboxyl group. To your left we have the NH2, which is the amino group. Then you have the hydrogen atom and you have your R, which is the side chain, and it is this R that will be changing. So you are not changing anything. This R is specific to different amino acid. So if you change this to CH3 now, it will give you alanine and the rest like that. Now, from this basic structure of amino acid, this side, the amino group, is reversed to as the N terminal. While this side, which is the carboxyl group, is referred to as the C terminal. Now, so for this method now, if this enzyme will cleave your protein and this chemical will also cleave, if they will perform their work, they will cleave these amino acids at the C terminal only. Cleavage will only occur at the C terminal of these amino acids. Now, in the next class, we will try and do some practical questions of protein sequencing, arranging amino acids of unknown protein to get a specific protein of interest. With this, we've come to the end of today's class. I hope you enjoyed this class so far. If you have any question, please drop them in the comment section and I promise to attend to every of your questions. Thank you. See you in the next class. Bye bye.